lab at Yale University as a postdoctoral fellow, where he focused on developing integrative frameworks and models for analyzing multi-omics data from the ENCODE and modular ENCODE projects. In 2012, Dr. Cheng established his own laboratory in the Department of Data Sciences at Dartmouth College Medical School. In 2018, he transitioned to the Department of Medicine at Baylor College of Medicine, where he continues his groundbreaking work in computational oncology. He has received numerous awards, many papers. The floor is yours, Professor.
All right, can I start?
Yes.
Okay. Yeah, thank you very much for the invitation and the kind introduction. Today I will talk about my work in the past 5 years. So this talk is focused on how do we develop new biomarkers for predicting cancer patient prognosis or response to a different type of treatment, especially immunotherapy, by using the tumor intrinsic or tumor extrinsic features. So the reason my lab focuses on lung cancer is because cancer is the leading cause of death, is the second most common cause of death in United States and probably across the world. And every year in United States, there's about 1.9 million of new cancer cases, and more than 600,000 cancer deaths in the United States died from cancer. If you calculate like how many cancer cases per year is more than 1,600. So in the whole life of a person, everyone have about 40% of chance in the lifetime to be diagnosed with cancer. So that's a pretty large proportion. But luckily, the reason in the last decades, the revolution. The cancer treatment is revolutionized by the development of a new type of treatment, the immunotherapy, for example, especially the immunocheckpoint blockade therapy have been used for treating different types of cancers. And these slides, I show one example. You can see this is a clinical trial result by using the anti-PD-L1 immunotherapy for treating non-small cell lung cancer. You can see here, like there are two arms in this trial. The immunotherapy group showed significant improvement in the survival, in the progression-free survival, compared with to the control arm treated by chemotherapy only. So there's a significant improvement in the long-term benefit by using the immunotreatment. However, so like only on average, there are only about 30% of patients will receive durable benefit from treatment, even in the promising cancer type. Like we know that the melanoma, lung cancer, this cancer type with high level of somatic mutations, and therefore there tend to have a hot immune system, immunotumor microenvironment, and therefore more likely to be responsive to the treatment, to the immunotherapy. However, even for this cancer, there's only about 30% of people who will receive long-term benefit. And you can see for other cold cancer type, the response rate can be quite low. And another information we can see from this figure is that if you look at the second one, the colorectal cancer with microsatellite instability, these are the patients that's highly sensitive to the treatment. That means by using some biomarkers, you can improve patient response rate to immunotherapy. And in this talk, I will talk about how do we develop prognostic biomarkers and predictive biomarkers to direct personalized treatment in cancer. What is prognostic biomarker? It is something that you can using the biomarker to determine which patient will have longer survival, which patient will have poor survival on the standard care, on the standard treatment. So on the other hand, the predictive biomarker is the biomarker can tell you which patient will be more sensitive to a particular treatment. For example, so we can using the predictive marker to identify to select the patient that are sensitive to a particular treatment. The treatment can be chemotherapy or anti-PD-L1 immunotherapy. Okay, so my lab is focused on this one. So there's many gene signatures or biomarkers of different type have been proposed in cancer. So one of the example is one of the most successful example is Oncotype DX, which is a 21 gene signature that have been developed about 20 years ago. It is both prognostic biomarker and predictive biomarker. That means it can select the patient with longer survival for the ER positive, HER2 negative early stage breast cancer patient. It can also predict which patient is sensitive to the chemotherapy. Okay, that is a very successful gene signature that have been used in clinician and have been in the market for direct treatment of breast cancer. And today I will divide my talk into 3 parts. In part 1, I will talk about the biomarkers developed from tumor intrinsic features. In part 2, I will focus on the tumor extrinsic features for developing biomarkers. Third one, I will talk about some new technology and how do we utilize these new techniques to develop biomarkers. This technology including CosMx, the spatial transcriptomic data, or single cell image data such as image mass cytometry. I will start it from part 1. Yeah, when we talk about the biomarkers, so basically we are interested the features that are correlated with patient prognosis or correlated with patient response to immunotherapy. When you look at a tumor, we can roughly divide the tumor into 2 parts, the tumor intrinsic part and the tumor extrinsic part. When we talk about the tumor intrinsic part, we talk about the genetic changes, the genomic changes, or the epigenetic changes that occurred in the tumor cells. Okay, that will change the pathway that driver tumorigenesis or driver tumor progression. So these are basically the intrinsic features. On the other hand, you also have like the tumor microenvironment, including the immunocells or stromal cells in the tumor, surrounding the tumor cells, right? This forms the tumor microenvironment. These features are very important for determining cancer patient prognosis, and especially as you can imagine, they have strong impact on patient response to immunotherapy because they're provide basically the tumor immuno environment. And in this talk, in part 1 especially, I will start it from tumor intrinsic features. Okay, so I will start from a very simple example, and we know that like p53 is a very important gene, very important tumor suppressor, like in all kinds of tumors. Here in the figure, I show the percentage of patients of different cancer type that carry the non-synonymous mutation of TP53 gene. However, when you separate the patient based on the p53 mutation status, you will see that the p53 mutation status of itself is not prognostic. Like as shown here, there's no significant difference between p53 mutated and TP53 wild type patient. They have similar survival. And so this is kind of counterintuitive. Why this important driver gene is not correlated with patient prognosis? So we think there are many reasons, because it is essentially the inactivation of the p53 pathway that's driver the patient the tumor development and prognosis, and determines the prognosis of the patient. Even though the TP53 somatic mutation is the most dominant mechanism that inactivate the pathway. There are many alternative mechanisms that can also inactivate the pathway. So for example, the promoter hypermethylation of the gene, or deletion of the gene, or mutation of some other genes that's related to this pathway. All of them can inactivate the pathway. Therefore, if you just look at the p53 mutation alone, you will miss the other mechanisms. So based on this idea, so we just simply integrate the somatic the TP53 mutation information and the gene expression information to develop a gene signature that can predict the p53 pathway activity. I mean, I note here like it can not only select the patient whose p53 pathway is inactivated by the p53 mutation, it can also select the patients that are for which p53 is inactivated by other mechanisms. I can show you here. So first of all, we see the by using the signature, we can apply to gene expression data. So if you have gene expression data for like this is the lung cancer data I'm showing here, and you can calculate the p53 signature score for each of the patient. And we see that like patient with knowing p53 mutation tend to have higher signature score than the p53 wild type. That's the reason why we call the signature score as p53 deficiency score. A high score indicates the inactivation of the pathway, right? So when the score is high, that means the pathway is more inactivated with lower p53 activity, right? So here we show that again, like if you stratify the patient based on the p53 mRNA expression, you don't see significant difference between the high expression group and the low expression group. And in D, we show that p53 mutation status is not working. It cannot distinguish good and versus poor prognosis. However, the PDS score like can stratify patient into two different groups with high prognosis and poor prognosis, as shown here. So therefore, the signature overperforms the p53 mutation status for predicting patient prognosis. And this prognostic value is independent of other clinical factors. That means when you use multivariate Cox regression model to adjusting for other clinical variables like age, tumor stage, or smoking status, and the score is still correlated with patient prognosis. That means the score can provide additional prognostic value for prediction prognosis. And when you do the stratified analysis, when you focus on your analysis using the stage only samples or stage 1 samples or stage 2 samples, the PDS score is still prognostic. So encouraged by this observation using the p53 simple example, we extended this idea into a framework. We call this GASPAS. The idea is like because each patient may have multiple somatic mutations. Some sample can have both p53 mutation and EGFR mutation in the non-small cell lung cancer, and we develop a framework that's identify for each gene. We determine how this gene's expression correlates with the presence of different type of driver mutations. The mutation can be somatic mutation, like the TP53 mutation. It can also be amplification. For example, the MYC amplification in some samples. So we using a model, we can consider all the driver chain aberrations in a simultaneous manner. And this framework will define a signature for each of the driver genomic aberration event. So for a cancer type, you can have maybe 40 or 30 something different signatures. Each of the signature will characterize a particular driver genomic aberrations, and basically it reflects the activity of the underlying pathways associated with this driver event. And finally, you can apply this signature to a novel gene expression data. You convert the gene expression matrix into a signature multiply sample matrix, and then you can use this matrix to correlate it with patient clinical phenotype and identify the signatures that are related to each of the phenotype. For example, you can predict prognosis, you can predict patient response to the treatment. So again, we tried this framework first in lung adenocarcinoma, and we're using the TCGA data to define the gene signatures. And then we apply these signatures to independent data from previous lung adenoma microarray dataset or RNA-seq dataset to show their clinical values. So basically, from TCGA, we select 23 genomic events that represent amplification of a cancer genes or deletion. There are 3 genes that are frequently deleted in lung adenocarcinoma, and there are also 10 genes with frequent somatic mutations in lung cancer. And in total, by using the framework, we defined 36 gene signatures. And on the right side, that just using the TCGA data, we show that like the gene signature score are more prognostic compared with the driver aberration event alone. You can see here, if you use the original mutation status, none of these 36 events are associated with prognosis. However, by using their corresponding signature, you have multiple signatures that are associated with patient overall survival. And then we tested this data in previous publications. I mean, here I select one microarray data that's published many years ago. So they're including a total of 200 lung adenoma patients. So we just simply calculate the signature score in each of the patient. You got a matrix, and we just simply do the clustering analysis. And you can see here, based on the signature scores, is divide the patient into 2 categories. So I showing here like the red one and the blue one. So when you compare the prognosis of these 2 patient clusters, you will see that the red cluster, the red group, tend to have a longer survival than the blue group. That means these signatures can basically separate the patient into different prognostic groups. And the right side shows the prognostic association of each individual signatures. We find that many of the signatures are correlated with patient overall survival, as shown here. So I will not cover all signatures, but I'm using a few signature as an example. One of the example is EGFR signature, and EGFR is a very important mutation in lung cancer. There are already 3 generations of drugs that are designed to target EGFR mutation to treat patient. And you can see here, like basically we find that the EGFR signature score is correlated with the EGFR pathway activity. So in A and B, we select the EGFR, the phosphorylated EGFR protein at a tyrosine. The location is in the 1068 amino acid. Is phosphorylation level is indicate how active the EGFR pathway is. You can see here, like the EGFR score in both TCGA tumor samples and CCLE lung cancer cell lines is highly correlated with the measured the experiment measured EGFR pathway activity. And the EGFR score is also correlated with the patient sensitivity to EGFR inhibitors, as shown by erlotinib and gefitinib, as shown here. You can see there's a significant correlation between EGFR score and the patient sensitivity to the EGFR inhibitors. And more importantly, even in the wild type, like EGFR wild type, we know that like EGFR mutant cell lines or patient are more likely to be sensitive to the inhibitors. However, you can see here, like even in patient or the cell line without the EGFR mutation, the score still provides some values to distinguish the sensitive cell line or the sensitive patient. So that means by using the signature, you can direct the treatment of using EGFR targeted treatment. Then we apply the signature to clinical trial. The first one is a small data. There are only 10 patients, and we calculate the EGFR signature score in this 10 patient and basically divide them into two groups with high score, 5 patients, with low score, 5 patients. Their survival is different. It's significantly different, even though there are only 10 samples. And the signature is still correlated with the survival after adjusting the clinical variables. In another clinical trial is the first one is treated by EGFR inhibitor gefitinib. Another one is treated by sorafenib. Is from the battle trial. There have many more patients here, like 21 responders and 14 non-responders. And you see here the EGFR score tend to be high in the responders. I mean, in the other hand, in other words, that means patients with high EGFR score are more likely to be responsive to the treatment, to the inhibitor. And this we can quantify the accuracy by showing the raw curve. So using the score, you can achieve classification accuracy of 0.79, right? And you can also show that like the signature score is associated with long-term benefit from the treatment, as indicated by the survival analysis. All right. So more importantly, remember, like we can actually calculate the EGFR signature score for each of the patients. Oh, go ahead.
On your screen it says weekly meeting with Chiang at the bottom right corner. Would you mind dismiss?
Oh, you mean?
Yeah,
yeah. Yeah, that is.
Just dismiss that, please.
Okay.
Excellent. Thanks.
All right. Good. So we can calculate sample specific or patient specific signature score, and we know that each patient have different type of somatic mutations. So we can actually rank the somatic mutations based on their EGFR score. And for example, here, these two, like you can see, they have high mutation frequency in the lung cancer patient. So these are FDA approved target. So patients basically have these two type of mutations are knowing from previous clinical trial to be responsive to the EGFR inhibitors. The others, there's no enough clinical trial data to support their effectiveness. However, like that depends whether the oncologist will make the treatment or not. But you can see here from our calculation, even some of the rare mutations, they have high EGFR score, which means these mutations can also result in overactivation of the pathway, and therefore patient with this mutation should be responsive to the treatment. On the other hand, on the right side, you will see that like there's some mutations with very low EGFR score. These mutations might not be responsive to the EGFR treatment. So to test this, maybe we need a larger sample size. For example, from the Tempest or from Caris, there already have millions of samples to test this kind of things. And also like in lung cancer, EGFR is involved in two genomic aberration events, EGFR somatic mutation and EGFR amplification. So very interestingly, like we find that these two mutations have very different effect on prognosis. They also have very different effect on the tumor microenvironment, as I'm showing here. The EGFR mutation score is associated with a better prognosis. That means high EGFR mutation score. Usually, the patient have a longer survival. However, high EGFR amplification score is associated with a worse prognosis. And we also find that the EGFR mutation score, but not EGFR amplification score, is correlated with patient response to chemotherapy. As we're showing here, you can see like the mutation score. When we have data from the chemotherapy trial, so only the EGFR mutation score is correlated with the response status, but the EGFR amplification has no correlation with patient response status, as shown here. And finally, their association with the hotness of the tumor environment is very different. High EGFR mutation score is typically associated with high level of immunoinfiltration, as shown here. But the amplification score tend to have low correlation or negative correlation with the tumor microenvironment. That means like lung cancer patient with EGFR gene somatic mutation tend to have hot immunoenvironment. However, with amplification, EGFR amplification, that tend to have a cold immune environment. That probably indicates this patient may have different sensitivity to immuno checkpoint inhibitors. That's a story about EGFR. We also have another signature, STK11. We find that this is the only signature that's correlated with patient response to immuno checkpoint inhibitors. So this data is from, as we're showing here, if you look at the non-small cell lung cancer patient under standard treatment, it's not correlated with survival, right? It's not a prognostic. However, when we applied the signature to a clinical trial, which is Patil, the first author is Patil, is from the OAK clinical trial, and you see that like patient that are responsive to respond to PD-L1 treatment tend to have lower STK mutation signature scores, and survival analysis indicate that like these patients can receive long-term benefit from the anti-PD-L1 therapy. And I find some literature there already have some trial to test by using the STK11 mutation status to pre-select the patient for ICI treatment. But here we find that the signature maybe provide a better prediction accuracy compared with the mutation status alone, following the same idea as the p53 pathway story. And therefore, I summarize my part 1 presentation. The driver gene signatures can recapitulate the pathway activity underlying the driver mutations, and therefore there can be more prognostic and more correlated with patient clinical outcome. And number 2, some of the driver gene signatures is predictive patient response to specific treatment. I show one example of chemotherapy, one example of immuno checkpoint inhibitor treatment. And finally, the signatures related to the same gene, for example, the EGFR mutation signature and the EGFR amplification signature, they have very different patterns in terms of correlation with clinical features or immunological features. Yeah, I guess that's the part of I want to cover in my part one. And if there's no questions, I will move on to the second part, the tumor intrinsic features. As I mentioned in the beginning, the tumor extrinsic features are those tumor marking environment, like including the immunocell infiltration and all the relative abundance of different
So one
immunocell
question
types.
came in
Oh,
since
yeah.
you asked. One question is how to use the gene enrichment pathways to get the gene signatures.
Gene enrichment pathways to get the gene signatures. Uh, in my presentation, in our study, we using the framework to develop gene signature to modeling the driver genomic event. We didn't start it from the enrichment pathway. However, if you have a good set of gene gene set, for example, like p53 genes, you may also be using the gene set enrichment analysis, especially single sample GSE analysis, to calculate a pathway score to do that. I mean, we did that previously. Many of the pathways is also associated with patient prognosis.
Thank you. Also, how to evaluate these driver genes score? Are these scores affected by these driver gene mutation frequency, gene expression, etc.? Thanks.
There's not necessarily a correlation between their prognostic value and their population level frequency, so mutation frequency. For example, there are some genes are more frequently mutated in a particular cancer type, but they are not necessarily to be prognostically relevant. However, some rare driver mutations, but rare, I mean, it's not as frequent as p53, right? However, there's still a significant proportion of cancer patients have that mutation. Like STK11, I guess, is only 10% of in lung cancer, and that one you can see like it have very good predictive value for immunotherapy.
Thank you.
Yeah,
Yes,
address. Okay. Now let's move on to the tumor extrinsic features. And so we know that like there are many gene expression data have been generated to profile the expression in the tumor samples. However, like when you have a tumor sample, it's not just the tumor cells. There are like depend on the tumor purity, right? There are many other cells like stromal cell and also immuno cells that's infiltrating into the tumor microenvironment. So it's a mixture of cells. So accordingly, the gene expression is not just reflect the gene expression in tumor cells. It's a mixture of gene expression of different cell types. So based on this concept, like there's many algorithms have been developed, like the CyberSort, the timer algorithm has designed to calculate to infer the immunoinfiltration based on the bulk gene expression data. So we actually, a few years ago, we developed an algorithm called Bayes algorithm to infer immunocell infiltration levels in the samples, and we apply that inferred immunoinfiltration levels to check to predict patient prognosis or patient sensitivity to immunotherapy. And again, like I select the lung cancer as the example. And so basically, we select a lung cancer patient with more than 200 samples, and for each of the sample, we apply the algorithm to infer the infiltration level of 6 major immunocell types, like including 2 B cells and 2 T cells, CD4 and CD8, NK cell and myeloid cell. And again, we started from the simple analysis, like clustering analysis. And you see here, like, so the simple clustering analysis divide the patient into 2 groups, with one group with more immunoinfiltration, another group with lower level immunoinfiltration. These 2 groups of patients have significantly different prognostic survival times as shown on panel B. And if you look at each individual cell type, you see that the naive B cell, the CD8 T cell, the CD4 T cell is associated with, is positively correlated with patient prognosis. That means high level of infiltration of these immunocell types are correlated with longer survival. On the other hand, the myeloid cell is negatively correlated with patient survival. High level indicates poor survival. Okay, and again, like this, the prognostic value of these immunocells is independent from clinical trial. When you're adjusting tumor stage, age, or smoking status, so all these immunocell infiltration levels is still significantly correlated with patient survival. Furthermore, they are supported by the stratified analysis by the stage. Like in stage 1, there remain to be significantly associated with survival, as showing for the three immunocell types. Okay, so this is another example. Remember, like we can using the algorithm to infer the infiltration level of immunocells. Here I choose CD8 T cell infiltration level, and because we are using the marker rate data, so we know the key immunocheckpoint genes like CTLA4, the expression level of the gene, right? So we combine the gene expression and the CD8 infiltration to stratify into 4 groups. And very interestingly, you see that like the blue group, these patients have high level of CD8 infiltration and low level of CTLA4 expression. These patients have very good prognosis. None of them have recurrency within 10 years. And the second group, which is the yellow one, even though they have high level of CD8 expression, but they also have high level of CTLA4, the immune checkpoint gene. These patients, about 25% will have recurrency within like 5 years. Okay, and the other 2 groups, the green one and the red one, they have a low level of CD8 infiltration, regardless of the CTLA4 expression levels. I mean, they always have the worst survival time, the shortest survival time as shown here. So that means like maybe the yellow group, the patient with high level of CD8, high level of CD8 positive T cell, but low level, but still have high level of CTLA4 is the most interesting group. Then because if we use anti CTLA4 immunotherapy to neutralize the immune checkpoint protein, and maybe we can push this group into the blue group, right? And therefore, there will have an extended survival time. Yeah, so we also extend the analysis to other cancer type and perform a pan cancer analysis. And I guess in more than how many, about 10 different cancer types, we find the prognostic value of the CD8 T cell infiltration for prognostic prediction, including glioma, melanoma, lung adenocarcinoma, and bladder cancer, as shown here. For the other cancer type, it doesn't mean the CD8 T cell is not associated with prognosis. So many of these cancer type, when we perform the analysis, they don't have data set with large number of samples, or there are or the survival time data is not in high quality. So, but at least for 10 cancer type, we see the prognostic value by using the inferred CD8 T cell infiltration. And we also do some analysis to see to show the predictive value of the immunocell infiltration inferred from gene expression. And for example, here we find a memory B-like signature is predictive of patient response to immunocheckpoint inhibitors. And this is here I show 2 independent data, the Snyder data and the Hugo data. I guess the first data is bladder cancer, but the second data is melanoma data. And you can see that like compare with patient who receive non clinical benefit, the clinical benefit patient they tend to have high level of memory B-like memory B-like cell infiltration, and it's also supported by the survival results. And in another data, you see there's a gradual increase of the memory B infiltration from a patient who receive progressive disease to partial response to complete response, right? And also the result is supported by the long-term benefit as shown by the survival analysis. And it's not only working for anti-PD-1 or anti-PDL1 based treatment, it's also working for anti-CDL4 treatment. This is the result from Van Allen's data. The patients are treated by anti-CDL4 therapy. And similar story, basically, patient who have high level of memory B-like cell infiltration at the baseline are more likely to have long-term benefit from the treatment. Okay, so remember in part 1, I talk about the tumor intrinsic features, and here I talk about tumor extrinsic features. And in one paper a few years ago, we tried to combine these two categories of features, and we found that by combining these two categories of features, you can further actually improve the prognostic prediction in breast cancer. So here in the paper, we intentionally select breast cancer to show the results because we want to using the famous Oncotype DX signature as a control. And you see that like by using Oncotype DX signature alone, you get an AUC score for distinguish good survival patient and poor survival patient. The AUC score is only 0.649. By combining the Oncotype score with clinical information like the tumor stage, the tumor category, cancer category, and you got an AUC score of 0.765. However, by using the tumor extrinsic features or the tumor and the tumor intrinsic features, we can easily get an AUC score of 0.8, 0.27. And like if you further incorporate the Oncotype DX score, you don't see additional increase. That means these features already covered the contribution from the Oncotype DX. But essentially, Oncotype DX capture the proliferation of the tumor cells, right? So by using these categories of these two categories of features, you already covered the information that's captured by the Oncotype DX. So here I show another example, which is a little bit different. So in a side project, we have interest on how the virus associated cancers related to tumor environment. So we know that there are at least 6 types of cancer types called virus associated with virus associated tumors, including liver cancer, head neck cancer, stomach cancer. And in that study, like we see how the virus infection change the proliferation of the tumor cell, the tumor intrinsic site, and how the viral infection affects the tumor microenvironment, right? So specifically how when you compare virus positive versus virus negative patient of the same cancer type, whether you see increased immunoinfiltration or decreased immunoinfiltration. So basically, I try to be short in this one. We find a very interesting story. I find the prognostic effect of the virus infection is determined by the balance between the two effects. So in one part, like in one side, the virus infection will generally promote the proliferation of the tumor cells. So there have some mechanism to increase the proliferation rate of the tumor cells. On the other hand, virus infection can also increase the immunocell infiltration in the tumor microenvironment, probably by generating virus antigens in the tumor site. And the balance determines how virus states correlate with prognosis. So as shown here in head neck cancer, we find that like the virus positive patient tend to have longer survival than the virus negative patient. So this is because like if you look at the proliferation of the tumor cells reflected by the MKI67, which is a proliferation marker. And you see that there's no significant difference between the virus positive and the virus negative patient, right? That means in this cancer type, the effect of virus infection on tumor cell expression is not that dramatic. However, you do see that the virus positive patient have high level of immune infiltration than the virus negative, and therefore you see like basically virus positive patient have longer survival. The other story is the bladder cancer. You see the opposite trend. Virus positive patient tend to have worse prognosis, as shown here. It turned out that the virus infection will significantly increase the proliferation of the tumor cells, even though it can also significantly increase the tumor immunoinfiltration in the tumor microenvironment, but the balance between them result in poor prognosis of the virus positive patients. So I guess I can conclude the second part. So the infiltration level of certain immune cells are correlated with patient clinical outcome. And by combining tumor intrinsic and extrinsic features, we can more correctly predict patient prognosis or patient response to immunotherapy. I think I only have about 10 minutes. I will quickly go through the part 3. And in part 3, like we utilize a recent published data, the image mass cytometry data, to develop a new type of biomarkers. As shown here, like this is a typical IMC data, IMC image for our area of interest. And we developed some algorithm to quantify based on the IMC data. Basically, IMC data tell you the spatial distribution of different type of cells, including immunocells in the tumor. And that tell you the position of each cell, the identity of each cell. So you can study for example, the relative distance between different cell types, and you can calculate the interaction between different cell types. So in one study, we develop a matrix to quantify the proximity between two types of immunocells, as shown here, and we investigated how the proximity between different pair of immunocells related to patient prognosis. Because in this data, they're provided IMC images for more than 400 lung cancer adenocarcinoma patient. That's a pretty good data set. And we rediscovered the previous reported results, like the proximity of T helper cell, macrophage, and B cell to cancer cells is correlated with patient prognosis. But more interestingly, when we extended the analysis to all cell pairs, we found that there are some cell type pairs are more correlated with patient prognosis. And interestingly, all of these, the top prognostic cell pairs are involved in M2 macrophage. So as shown here, like the proximity between M2 cell to the T helper cell can stratify patient into different prognosis. And then next we just see, so when people do analysis, they just see giving the IMC data, they calculate the infiltration level of T cells, B cells, macrophage cells in each of the sample, right? And they're related to the patient clinical outcome. So we find that the function of the lymphocytes may be affected by the macrophage. So we therefore stratified the lymphocyte, like for example B cell, T helper cell here, based on their proximity to M2 cells. So basically, instead of calculate a single overall TH proportion in an image, we calculate two values to quantify the fraction of M2 distal T helper cell and the M2 proximal T helper cells separately. And you see that very interestingly, we find only the M2 proximal the M2 proximal T helper cell is not correlated with survival. However, the M2 distal TH cell fraction in the patient is very significantly correlated with patient survival. Similarly, the B cell have a similar idea, and the TC cell is more interesting. Like the M2 distal TC is protective. The M2 proximal TC is actually hazardous to the patient. That's something like we think very interesting. And also, we applied a similar analysis. We're using the proximity score to study the cells, the interaction between different cell types. And we applied that to another larger scale data set in triple negative breast cancer. So there have clinical trial, there have patients from clinical trial that collect samples at the baseline during the on-treatment samples and also post-treatment samples. And here we focus on the baseline samples. And in their treatment, they have two arms. The first arm is treated by a combination of chemo and immunotherapy. The second one, as a control, is chemotherapy only. Then they're generated the IMC data. And we using the proximity score to quantify the strength of cell-cell interaction for all cell pairs. And we find the interaction between Treg and the cytotoxic T cell, like here, is significantly associated with patient response to immunotherapy. You can see here, their interaction strengths can classify responders versus non-responders with the AUC score of 0.717, which is much higher than what can be achieved by individual proportion, the conventional markers for predicting patient response. And you will see here, like when you compare the responders and non-responders, the strengths are significantly different. And more interestingly, the interaction strengths of these two cell types keep changing during the treatment. Like you can see the responders, their interaction strengths is keep decreasing from baseline to on-treatment to post-treatment. However, the non-responders, you don't see any change like in their during the treatment, like in the interaction strengths. And therefore, we propose a model. Let's say you can pre-select the patient whose baseline at the baseline level, you observed a strong interaction between Treg and cytotoxic T cells. And these patients are more likely to benefit from immunotherapy or combined therapy that's using both chemo and immunotherapy. Like currently, we are collaborate with clinicians to further validate these results in triple negative breast cancer and in lung cancer. Yeah, I will skip this part. Basically, like the IMC data is not available for, I mean, it's not as available as the gene expression data. So to address this issue, we actually defined some algorithm, like you can using the bulk gene expression data to infer the relative abundance of different immunocell types in each of the sample, and then you can you can determine, you can identify the immunocell pairs whose relative abundance is correlated with patient prognosis or correlated with patient survival. And we published this paper in about two years ago. So this is basically why we want to develop this. Like in melanoma, if you look at the prognostic genes or the prognostic cell types, you will find that the majority of cell type, the majority of immunogenes are protective to patient survival, to patient prognosis. The majority of immunocell types are protective. That's counterintuitive because we know that Tregs and many other immunocells are actually pro-tumor, right? That should be negatively correlated with survival. I mean, this is caused by the co-infiltration of these immunocells. So the TMGP, the algorithm is designed to address this issue. I guess limited by the time, I skipped this result. So basically, we can claim that by using the new advanced technology, we can develop new categories of biomarkers, and we can use computation method to utilize existing gene expression data to to improve biomarker development. So finally, I will thank all the collaborators and the grant that support this analysis. Thank you very much. I stop here.
Thank you for the fantastic talk, Professor. A couple questions came in. How do you study the immune cell interactions with the tumor cells into the cancer patients?
Can you repeat the?
How do you study the immune cell interactions with the tumor cells in cancer patients?
Oh, you mean the immuno interaction? It's like given the image mass cytometry data, so we determine that we're using the proximity score to measure the relative distance between all pairs of cells that fall into that two categories. So, for example, you can determine the location of all CD8 T cells and the location of Tregs, right? And you calculate their pairwise distance, and then you randomize the cell identity and recalculate the distance based on the permutative data, right? And you can do that many times, right? And then you normalize, you observe the data from the real image by using the non-distribution inferred from the permutative data, and you calculate the strengths, right? That strength tell you how strong of the interaction between that two type of cells, immunocells, compare with what we expected randomly.
Thank you.
Uh-huh.
For the genes that you have found between different cancers, is there an overlap, or for each cancer with an individual, they have different types of gene mutations?
The answer is yeah, there do have many overlapped mutations between different cancer types. And but like each cancer type also have their unique, like for example, like there's a gene VHL is uniquely mutated in renal cancer, but not in other cancer types.
How about the mutations in the metastasis cancers?
Oh, the other one is limited by the availability. So for example, in TCGA, the majority of data are actually primary tumor, right? There are only a small fraction of metastatic samples. In melanoma, we do see there have both primary cancer and local regional metastatic lymph node metastasis. But we didn't separate the primary versus the metastatic this one.
Thank
Even
you.
for TCGA, you know that the patient is stage 4, but the sample is collected from the primary side.
Thank you.
Yeah, but that's a very good question. So we can study if the data is available.
Thank you. Once you identify the genes, what's the next step? Maybe not by your group, by others. Once you know the gene is the mutated one, what do you do with that information?
So like currently, I guess at least in Baylor, the cancer center is collaborates with Tempus or Caris. So they'll collect the patient, and the company will do targeted sequencing of a panel of cancer gene for free, right? And then the clinician decides based on the mutation status, they decide whether patient will be sensitive to to a particular treatment. For example, like as I said, like if the lung cancer patient have the EGFR mutation, right, non-synonymous EGFR mutation, so it will very likely benefit from the EGFR inhibitors, right? So I guess our study provides some additional information. That means, so even for patients without the EGFR mutation, maybe there are a fraction of patients that can also benefit from the treatment because their EGFR pathway is overactivated by other mechanisms.
Thank you. One final question. There's a lot of movement in artificial intelligence helping many computational or other fields. Where do you see your field moving with the emergence of AI tools?
Yeah. For example, I introduced in my in part 1 by using the statistical framework to define gene signatures for each of the driver genomic event, right? So basically, you can make a AI version of that. Actually we are working on that. So using the TCGA data again, like you have the gene expression data, and you have the demethylation data, you have the like microRNA expression data, and on the other hand, you also have the mutation status of all these frequently mutated genes, right? And you can using all the encoders to generate embedding from each of the model of data, type of data, right? And you combine them, right? And then you can define a AI version of this framework. And if you just rely on like giving a macro data, you can maybe we can only use the gene expression data, right? Embedding from the gene expression data to calculate the mutation score of a particular event, right? And you can do the similar thing as what I presented today. So that will be the AI version of this framework. The rationale is the same. That means the genomic data can provide more accurate information about the pathway that drive the tumor development and progression. And therefore, you will expect that the predicted score from the AI will be more correlated with or more accurately predict the patient outcome than the mutation status alone. Same rationale, but different computational tricks.
Thank you. Thank you. One final question. What kinds of information could be important for the neoadjuvant treatment prediction?
Oh, neoadjuvant. So are you talking about neoadjuvant chemo or immuno?
Both. He didn't
Yeah,
clarify.
yeah. You can say like the latter part, that triple negative breast cancer data, image data, IMC data, is neoadjuvant chemo immunotherapy, is a combination therapy. So that's the only data like we showed, like we identify potential biomarkers that are predictive to the neoadjuvant treatment efficacy. I guess the barrier is there is only very limited clinical trial data that are released to the public. So I find some data that only give you the genomic data, for example, the gene expression data. However, the treatment outcome of the patient is not provided. So that way, you're not being able to develop the algorithm to develop to obtain the predict biomarkers. So I guess that's a barrier. But as long as there are data available, you can like using different ways to build up AI or other framework for developing new biomarkers. So we have very limited successful examples in this field.
Yes.
Not our
Thank,
fault.
thank you so much for the great talk and your time, Professor. Thank you.
Yeah, thank you everyone.
Thank you.
All right. I stop share.